So the whole unit 3 can be essentially divided into, give or take, 3 chapters. The first chapter deals with the basic concepts of microbial growth, the growth requirements, conditions, stuff like that. Second chapter is antimicrobial agents, physical or chemical ways to get rid of microorganisms. Very memorizable, very flashcardable. And the third chapter is about antibiotics. Same story, very memorizable, very flashcardable. Okay? So let's talk about basic concepts of microbial growth. So first of all, bacterial division, binary fission, it's an asexual division. There is no meiosis. There is no mitosis. So basically what you get, DNA is replicated, all the cellular components are doubled, and then the cell partitions in two. Okay? The only similarity to mitosis in eukaryotic cells is the formation of a septum that formed by the actin proteins, so annoying, formed by the proteins that is similar to actin, so-called FTSZ proteins. They essentially squish the dividing cell and separate two daughter cells. Does that make sense? Now, here's something to understand here. Come on, dude. Can I get rid of this? Okay, better. So something to understand. Cells double with every generation. Okay? So if you think about this, that every time cells divide, it takes a certain amount of time. We call it generation time. Sounds good? Generation time is not chiseled in stone. It's going to depend on external factors such as pH or availability of nutrients or competition or, I don't know, oxygen levels, you name it, temperature, doesn't matter. And this is my first question. Let's say you take microorganisms such as an E. coli and you grow it on an agar plate in the lab. Same microorganism can grow in your intestines in its natural environment. In which environment in the lab or in the intestines will E. coli grow faster? Okay, so hold on a second. So we have two versions, different people. One says intestines. What's your argument?
That's the environment it involves to go where.
Okay, it's a natural environment. Somebody said a lab. Why lab?
That doesn't have any competition against any other microbes.
And the Oscar goes to, yes, there is no competition in the lab. It has been shown that the difference can be staggering up to 6 to 9 times slower in the natural environment because there is a wild competition for nutrients. Okay? Now, there are going to be questions on the exam on calculating the generation time or the number of cells. So for now, I'm going to cut the crap. And I know that these questions are going to arise. I'm just going to go through them without mentioning anything on the slide. So let's say we have a hypothetical solution, okay, that contains 10 cells. Okay, make sense? Generation time is, what do you want it to be? Just give me a reasonable number, not like 37. Huh? Say again?
20 minutes?
Oh, come on. 7 minutes. The fastest one is 13 in the lab. Campylobacter jejuni. 20? Let's do 20. 20 minutes. Okay? Does that make sense? Yeah. So, how much time is needed to get to 2,560 cells? Okay? Does that make sense? Good? Okay. So how do we approach that? We say, okay, we do a table, generation and the number of cells. Everybody cool with that? So generation is 0. How many cells do we have at the start? 10, right? Okay? Everybody agrees with that? Yeah. That's like our starter. This is why we give number 0 to that generation, right? So at the first generation, remember, cells double. How many cells do we have? 20. At the second generation, how many cells do we have? 4. At the third one, 80. At the fourth, fifth, 320. 320, it doubles, you multiply it by 2 every time. 640, 1 to 80, and 8th generation 2560. Okay? So, how many generations do we have? What's the generation time? 20 minutes. 8 multiplied by 20 minutes, 160 minutes, or 2 hours and 40. Whatever you prefer. Does that make sense? Good? Okay. So let's give another example of a problem that can be on the exam. So I'm going to make it simpler. You see this little streaks? I don't know how to get rid of them. Start with 1 cell. Okay? Generation time, 30 minutes. How many cells in 3 hours? Okay? So, let's see how many generations we have. We have 3 hours. What is the generation time? Which is half an hour. So if it's half an hour for 1 generation, how many generations are we going to have over 6 hours? Oh, sorry, 3 hours. 6, yes. 6 generations. Good? So we have 1 at 0, 2 at 1, 4, 8, 16, 32, 64. That's your answer. Sounds good? Good? Okay. So, let's give another example. Start with $20 cells. I hope I'm not going to screw up on that, because I do quick calculations in my head. After 4.5 hours, you have 1 to 80 cells. So far, so good? What's the generation time? So, the approach should be the same. So we have, at the 0 generation, we have how many? 20. add the 1, 40, and the 2, 3, 4, 320, 5, 4, 640, and 6, 1 to 80. So how many generations do we have? 6 generations. How much time did we have? 4.5 hours, which is 300. Wait. No. 200. Help me out. 270. 270 minutes divided by 6 generations, right? Gives us 45 minutes. That's your generation time. Look, when I was in high school, calculators were banned in all classes that we took. No, you don't. Okay, we're good? So those are basically 3 types of questions that you can encounter regarding these calculations about doubling time or generation time, about number of cells, time, whatever. Clear? Okay. Let's get back to this. So obviously generation times vary widely. So the common question, classic question, if we would put 1 cell on the Agar plate and the generation time is 30 minutes, which is normal for E. coli, for example, in 24 hours by tomorrow, we would have about 1 pound of cells. We don't. Why? Why don't we have bacteria just swarming out of agar plates? Huh?
Not enough nutrients.
Exactly. Not enough nutrients. That's it. They run out of food. And probably produce enough waste to kill themselves. Okay? Good? All right. So this is a really important concept, the growth curve. I want you to understand what's going on here. So I'm not going to ask you, I promise, about logarithmic scale versus arithmetic scale. Just acknowledge that this is the weird logarithmic scale and just, take it as a given. Just don't want to waste time on math. So basically the idea is this. There are 4 stages of the growth curve in the colony of microorganisms that do not receive any input of nutrients or any removal of waste. Basically, you have your media, you have your bacteria, you seal it, stick it in an incubator, and observe. Does that make sense? So the first step is the lag phase. This is the time when bacteria adapt to the new environment. Okay? Good? Nothing happens, numbers don't change, they just figure out stuff. Then the most important phase, the logarithmic phase. In this phase right here, bacteria actively divide. The number of dividing cells is vastly greater than the number of dying cells. And the nutrients are abundant and wastes are very low. Does that make sense? At some point, more cells start to die and the amount of waste accumulates, right? So you enter the stationary phase, when basically the number of dying and dividing cells is equal and nutrients kind of lower, wastes go up. Okay? That's your stationary. And then if you don't feed your bacteria, if you don't add nutrients to the medium, they're going to enter the death phase. Okay? So number of dying cells is greater than the number of dividing. Nutrients are gone, wastes are increased. Okay, does that make sense? So I have a couple of questions about this. First of all, the populations in the wild, say your intestines or your skin or soil, in which phase do you think these populations exist? Stationary, of course, because either way it would be unsustainable. You have 3 pounds of bacteria in your intestines. If they would start divide, like you're gonna explode in a matter of few hours, okay? And same as with death phase. Another thing that I want you to consider, and that's kind of a food microbiology question. Everybody's over 21, I suppose? No? Okay, sorry. You can cover your ears. We're talking about alcohol. What is the normal strength of a table wine? Alcohol percentage of normal, like regular Cabernet, whatever. Huh?
14%.
Yeah, pretty much 12 to 14, yeah. Why? Why it's not 25? Why it's not 25? Huh? Why it's not like 20? Why can't we just like say again? But okay, think about the answer is in the growth curve. I'm dead serious. The answer is in the growth curve. Why it doesn't go to like? Why can't we just mix malt and some bacteria and get whiskey? 45%. Why can't we do that? What is alcohol essentially? Product of, so it's a bacterial poop, It's a waste that they produce, which kills them. Does that make sense? There are wines that have alcohol percentage over 20. What's the difference? What do you give those bacteria extra? Give them sugar. Yes, like really, really sweet grapes produce really, really sweet wines like Harris or Madeira because they have more sugar. Does that make sense? Now, another important implication for food microbiology is this: persisters. So you can send a population of bacteria into a death phase. You can give them antibiotic, or you can treat them with the toxic chemicals and they're going to start dying. Make sense? And if you do that, some of the cells will dramatically slow down their metabolism so they will be able to survive through that environmental onslaught. Does that make sense to you? So when, for instance, you take antibiotics, you take them for quite a long time. Like you have a strep throat, you're probably going to take it for a couple of weeks. And you should take the entire dose over 2 weeks. Why? Because these antibiotics essentially have a scorched earth strategy. They kill everything, including persisters. It just takes longer. If you stop early, persisters will survive, will rebound, and you have repeated infection. Now, food microbiology story. Do you buy pre-washed greens? Okay, fine. Why? Not quite. The greens that are sold as pre-washed greens, they are not just washed with water, they washed with a weak bleach solution. The key word is weak. It kills a lot of bacteria, but it doesn't kill all of them. And it sends a bunch of them in a persister state. Okay? So when you mix your salad and you eat your salad and these persisters end up in your intestine, they're like, oh crap, that's phenomenal. We're just going to grow back to normal. And that's what happens. So indeed, FDA recommends to rinse your pre-washed, quote unquote, greens at least twice, I think, if not 3 times. Okay? So I personally just take the risk. Okay? So, cell counting. I can tell you what I will ask. I will ask you to match the technique in its description. Okay? And I will ask you to identify whether a technique can count live cells or it counts all the cells whether they are alive or dead. Okay, so a direct cell count, I'm not going to read the description, you can read it yourself. So basically you put the sample on the Petrov Hauser chamber, stick it under the microscope and manually count the cells. It can distinguish live and dead cells. Okay? Make sense? It's not very straightforward, but it is doable. Coulter counter. So what you have here is a bunch of cells coming through what's called an aperture, the opening. And when a cell crosses the aperture, it changes the conductivity between two electrodes. Basically, the cell is registered. As you can imagine, it's just a flow, so you can't distinguish live and dead cells using Coulter counter. I have like question that is unexpected even for me. During which phase of the growth curve is the Coulter counter method reliable? Say again? The logarithmic, yes, of course. Because during logarithmic growth, you don't have a lot of dead cells. Does that make sense? So yeah, it's pretty reliable there. And it works only with the diluted suspensions of bacterial cells, because you can imagine that you have a really concentrated, more than one cell can cross the aperture and it will be registered as one. Now, this is the viable cell count. Basically, you have a sample and you make serial dilutions. We're going to talk about serial dilutions probably. Well, we'll see how it goes. Maybe even today. Okay? We're going to do some serial dilutions problems. Great way to calculate the actual number of live cells in your sample. I did the calculation once, how many agar plates it takes to do a proper growth curve for like a span of 12 hours. We got over 1,000. So it's a lot of work. The viable cell count it's a shitty job. Like seriously, it's not fun. Okay? Now, two last methods. I'm gonna start with the bottom one, the turbidimetry. So remember, yeah, that actually should be a different color since I use different. Okay, so turbidimetry is the measurement of the cloudiness of your sample. Okay, it encounters all the cells, dead or alive, doesn't matter, but it is a very useful method because what you can do a viable cell count and run a turbidimetry experiment at the same time. Does that make sense? And you can calibrate your turbidimetry. You can calibrate the measurement of the cloudiness and match it to the actual number of the cells. And at least for logarithmic growth phase, you're going to have, okay, if we have optical density of, 0.3, it means that we have concentration of 5 million cells per mil. Am I making sense? Okay, so you can calibrate your turbid. It's very convenient in the short term. And my favorite, most probable number. So, anyone swims in Lake Erie? I'm not judgmental, I do. Okay, which beach?
Edgewater, Lakeshore.
Don't do Edgewater. Don't. No. The only. No. Which one did you say? Where's that? Probably Bay Village is probably safe. I'm from the east side, so Mentor Headlands. That's the only one. Geneva depends. Anything else is a complete. It's a dumpster. It's a complete dumpster. Edgewater is the worst. Euclid Beach is not that far. Fairport Harbor is pretty horrible as well. We test it every semester. It's bad. Not in here. We don't have this lab here. Anyway, so how do you do that? How do you test the water? If you take 200 microliters of water and put it on the agar plate, chances are you're not going to catch any microorganisms, right? So you need a large volume. So here's what you do. Well, first of all, you can filter, but it's not up. That's what we do, but it's not quantitative, really. So what do? You get a bucket of water from a lake, and you have 3 sets of indicator media here. And you inoculate first set with 10 mils of water, second set with 1, and the third set with 0.1. Okay? Obviously, chances of microbial growth in the tubes that receive 10 mils of water are much greater. Everybody agrees with that? So you 24/48 hours later, you look at your samples, and here's what you see. You have certain number, all 5 of them, in 10 mils, only 2 in 1 mil, and nothing in 0.1. Then you literally, and I saw that book. You literally go to a special book, probably now it's like online database or whatever. You go to a book, you open the table, and it tells you that if you have a distribution of 5 to 0, okay, it means that the most probable number of cells in 100 milliliters of your sample is 49. Now, I want to highlight most probable number. So, like, I look at the class and I say, well, the number of Cleveland Browns fans in that room is around 80% because there are 20% of people who don't care about football, including me. Okay? It's unlikely that nobody likes Cleveland Browns. It's unlikely that everybody does, right? So the idea of most probable number is this. The most probable number that you have in 100 mils of your sample is 49. But the actual number lies between 17 and 130, the confidence interval. Does that make sense? Is it possible that you actually have 18? Yes. Is it likely? No. Is it possible that you have 128? Is it likely? No. Does that make sense? The whole this is like when you hear that, such and such state park or beach or whatever are deemed not safe for swimming, this is what they did. Most probable number method. Okay. Now, obviously, don't memorize the percentages. I mentioned that before. There are a few things I want you to know definition-wise. Essential nutrient/growth factor, any chemical that is required for a microorganism to grow. Sounds good? Anything from iron to amino acid tryptophan. And this. I can ask you, where can you find nitrogen? Proteins, DNA, or RNA? Does that make sense? Or which elements comprise phospholipids? Phosphorus, carbon, hydrogen, oxygen. Fair? Okay, now, this is my favorite and students' least favorite part of that unit. Okay, biogeochemical cycles. So we're going to talk about 3 cycles, how different elements well, cycle in the biosphere and atmosphere and lithosphere. And what is the role of microorganisms in it? Sounds good? Okay, so first we're going to talk about carbon. So two biggest parts of the carbon cycle are heterotrophs and autotrophs. So heterotrophs run cellular respiration, okay? So in this direction, that's going to be heterotrophs. Make sense? So they oxidize organic carbon, forming CO2. Autotrophs then use that CO2 and water to form organic compounds in the process of photosynthesis. Does that make sense? So this is pretty much like 2 steps, but it's not just 2. There's some more. So there are microorganisms called methanogens. So they use anaerobic respiration, and they use carbon dioxide as a final electron acceptor. Okay? And they produce the gas called methane. It's going to be a weird question. Did you ever drive by the landfill? No? If you ever happen to drive to Cincinnati on I-71, once you go beyond Columbus, I did that route more than I ever wanted to. Probably 10-15 miles south from Columbus on I-71, there's a pretty large landfill on the right-hand side of the highway. If you ever drive through Bedford Heights, Bedford, they have a large landfill there, already closed. What you're going to notice is that there are pipes sticking out of the landfill. Landfill is an extremely anaerobic environment, but carbon dioxide from the air can easily penetrate in there. And this carbon dioxide, along with the carbon dioxide that is produced during certain types of fermentation, will be used as the final electron acceptor by bacteria there to form methane, and those pipes let methane out. If you don't do that, it's going to be a lot of fun. In my hometown, several buildings were built on the old landfill without any pipes. And once a month, one of the buildings exploded because of the methane that was leaking from the landfill. I'm not making this shit up. Like, we had it in the news. Like, roughly once a month, methane. So that's not the main issue, though. The problem with methane and CO2, both of them are greenhouse gases. So they significantly contribute to the global warming. And CH4, methane, contributes even more than CO2. So, well, first of all, ideally, this methane should be captured, it should be burned to use as energy, whatever. But we don't do that. Well, sucks to be us. Anyway, we can use methane as the carbon source in the reactions of oxidations like this. Remember, this is not a chemistry class. I'm not going to ask you about the reactions. I want you to give me a conceptual understanding, like, what do heterotrophs do? They oxidize organic carbon to CO2. What do autotrophs do? They reduce inorganic CO2 to organic compounds. What do methanogens do? Okay, they produce methane. What do methanotrophs do? They use methane as the carbon source. Okay? Awesome. Nitrogen cycle. Super exciting. So nitrogen is an extremely inert gas. It's, I don't know about other languages, but Russian name azot comes from the Greek that says, do not support life. No shit. Anyway, nitrogen is a very inert. but it is required in biological molecules such as proteins, nucleic acids, whatever, so you have to snatch it from the air somehow, right? So the first step in the nitrogen cycle is, nitrogen fixation. So what you do, you take nitrogen and you convert it into ammonia. Microorganisms that are listed here, you don't need to memorize them. During nitrogen fixation, they do it at the ambient temperature and normal pressure. Humans do similar reaction at not exactly the same, but they do it at about 500 degrees Celsius and 400 to 500 atmosphere pressure, okay, with platinum catalyst. Then you have this, so bacteria are much better than us. Once they produce ammonia, ammonia is incorporated into organic compounds in the process of aminification. Okay? Good? First comment, sort of relevance of this. If you do gardening, like vegetable gardens, especially if you grow potatoes, plant potatoes next to beans. Beans have rhizobium on the roots. Rhizobium can fix nitrogen from the environment, enriching the soil in nitrogen. And we did that when I was a kid, more than I wanted to. But the potato harvest is enormous, just fantastic. Okay? So now, once this ammonia was incorporated into organic compounds, eventually living things die, and they release ammonia in the environment. And ammonia in the environment is oxidized to nitrite and nitrate eventually in a process called nitrification. I probably should use a different color to highlight specifically that term, nitrification, all right? So this is also important because in that oxidized form, nitrogen is also available for the plants to grow, to use and grow, okay? This is why we use nitrates as fertilizers. Now, here's one problem with nitrates. Are you familiar with what's that effect on the lake when algal blooms? Ever heard about that? Algal blooms? No?
It's a big problem.
So it's a huge problem. So it's a huge problem in Lake Erie because people would, farmers would obviously put fertilizer in the fields, and then there's rain, fertilizer runs off into Lake Erie, and suddenly all this algae have an enormous supply of available nitrogen in the form of nitrates, and they just grow, okay? But the problem is those algae grow for a very short period of time. They grow and they die. And when they die, decomposers decompose them using oxygen from the water. And water becomes eutrophic and... No, not eutrophic. No, I forgot the term. But anyway, oxygen is sucked out of the water, which results in the death of fish and other lake wildlife. Okay? It's a huge problem. So, what do you do about it? You do denitrification. So basically you catch that agricultural runoff and you literally run it through the filters with bacteria that will convert nitrate back to nitrogen. Go ahead.
Once it's already in the water, can you do that?
Say again?
Once it's already, once the nitrogen...
Oh, you mean when it's in the Lake Erie? So the question is, can you do it once it's in the Lake Erie? No. Volume. Just because the volume. This, unfortunately, yeah. So you have to catch it. And bacteria and archaea and protozoa are heavily involved in the water recuperation process. There is an actual layer in the filtration system called Schmutzdecker that contains all different microorganisms that are quote-unquote tasked with certain bacterial recuperation matters. Okay, sulfur cycle, and pretty straightforward. So you've got hydrogen sulfide, which is a reduced form of sulfur, and the reduced form is oxidized. Can be photosynthesis, anoxygenic, or certain chemoautotrophs. It is oxidized to mostly sulfate. And then here's the deal. Sulfate, think about this. This is ingenious. So you have anoxygenic photosynthesis. Photosynthesis that does not produce any oxygen, right? It goes from H2S to sulfate. Then you have anaerobic respiration. I'm going to highlight anaerobic respiration, in which sulfate is the final electron acceptor, and that produces H2S, so we're back to the square one. That makes sense? And the cycle goes over and over and over. There are some, rogue species that can convert between hydrogen sulfide and sulfur, just like molecular sulfur and vice versa. But the point is, H2S is really important. The availability of hydrogen sulfide is really important because proteins require sulfur. And you can only incorporate sulfur in the organic compounds if it's in the form of H2S. Okay? Now, full disclaimer. What do I want you to know? I want you to know like the concepts, not the formulas. So if I tell how can we get from hydrogen sulfide to sulfate? And I will be happy to provide you with formulas. You tell me, anoxygenic photosynthesis for the most part. How can we get back from sulfate to hydrogen sulfide? Well, anaerobic respiration. If we get back here, what happens during nitrification? Well, ammonia is oxidized to nitrate eventually, right? All good? Okay, so communities. I'm not going to spend too much time on rereading the table here. I'm going to ask to match the definition and the term. Full disclaimer, I can give you a definition like what's going on in commensalism. There are two organisms. One benefits from the interaction and another one is neither harmed nor benefited. Sure, I can give you that. But I can also give you a question like this. So imagine I have two microorganisms, right? Each of them A and B. Each of them can grow independently. Sounds good? But when you put, totally fine. But when you put them together, the growth of each of them is dramatically increased. So you look at the list, synergism. Okay, does that make sense? So they benefit both, but it's not necessary for the survival. So I will give you both types, purely definition and hypothetical situation. Okay? Now, understand the difference between planktonic and biofilm form. What you have on your tables in those tubes is a planktonic form. What you have on your teeth is biofilm. Understand the basic steps of biofilm formation. So you start with primary colonizers that produce so-called extracellular polymeric substances. What they're for? They protect against desiccation and predation. They're also sticky. We, in the last unit, we were talking about Streptococcus mutans, if you remember. It's a microorganism in your mouth that uses glucose to form carbohydrates, this sticky layer that protects it and attaches it to the surface of your teeth. So it can survive there, I guess, and use fructose as a source of energy, producing lactic acid, which will destroy your enamel and that's not good. Okay, does that make sense? Now, these extracellular polymeric substances, in addition to protecting against desiccation and predation, also quote-unquote attract secondary colonizers, so they start sticking better, and eventually your biofilm reaches critical density. My favorite analogy for critical density is this. Imagine that you are like in some unexplored land, nobody's around you, and you're just building your house, okay? You can do whatever you want. Like you can shoot fireworks, you can walk around naked, nobody cares, you know? But more and more people move into the area, and now you have to interact with the neighbors, so your communication changes. And at some point you have some like laws and shit like that. So this is critical density. When the population of bacteria in the biofilm reaches critical density, the level of quorum sensing molecules skyrockets. Quorum sensing refers to the interaction between bacteria and a biofilm. So they start coordinating their activities. I will not ask you to distinguish this types of quorum sensing molecules for gram negatives versus gram positives. I want you to understand that quorum sensing molecules enable microorganisms in the biofilm to communicate and to change the metabolic activities, producing virulence factors, or do other responses in concerted fashion. Be clear? Go ahead.
So if you detected quorum sensing molecules, you would know that your sample is past the critical density
Yes.
point. Okay.
Again, detection, it's a good question. The question was if you detect quorum sensing molecules, you know that it's past critical density. If you detect it at a certain level, okay? And they are autoinducers, so once bacteria reach that critical density, the level of these molecules skyrockets. So it's sort of an exponential curve rather than a linear one. Now we're going to talk about the requirements for growth. I'm going to spend some time here. So, first of all, what can I ask you about this? 3 types of questions. Question number 1. This type of microorganisms does not use oxygen for their metabolism, but can survive in the presence of it. Air-tolerant anaerobe. Does that make sense? I'm going to ask you to match a term and the formal description of oxygen requirements. I'm going to skip obligate aerobes and anaerobes, obviously. Well, pretty straightforward. So another example I'm going to give you. Helicobacter pylori requires a very specific amount of oxygen for growth, and this amount is around 5%, which is much less than the amount of oxygen in the air. What type of microorganism is that? You think, okay, very specific amount, lower than in the air, microaerophile, Goldilocks, okay? And finally, another example, Staphylococcus aureus, in the presence of oxygen, runs cellular respiration, aerobic respiration. In the absence of oxygen, though, it can switch to fermentation and behave like an anaerobe. What type of organism is that? So you think, okay, does it have to ferment? No. Can it, though? Yes. So it can ferment facultatively. It can survive without oxygen facultatively. Facultative anaerobe. That makes sense? I'm not telling you that I'm only going to ask about those 3. I just gave you an example. So it can be a hypothetical situation, it can be a pure definition, or something similar. Now, I also can ask you about this. You look at the tube and you discover the growth at the very bottom. What is that? Obligate aerobe, okay? Does that make sense to everybody? Right, perfect. Now, I'm going to talk about the bottom page. You need to know the 3 enzymes. There will be no questions on any of the reactions per se. I'm not going to ask you what are the reactants, what are the products? No, God forbid. But I can ask you this. which enzyme neutralizes hydrogen peroxide? And you need to identify that, either peroxidase or catalase, whatever. Does that make sense? The hardest part here, this is why I spent so much time on the slide. The hardest part here is this. So when organisms respire, they produce reactive oxygen species. That's inevitable. We do that, bacteria do that. And 3 enzymes that neutralize them are in the bottom table. Superoxide dismutase, catalase, peroxidase, right? Now, if you start thinking about the oxygen requirements, realize that they are inextricably linked to the presence or absence of those enzymes. Think about this. Obligate anaerobes, they die in the presence of oxygen. They cannot neutralize any of that stuff. This is why, they quote unquote choose to live without oxygen, because without oxygen, they don't have to. Okay? When you look at obligate aerobes, they have all of them. Okay, sounds good? But why do we pay so much attention to this? Well, so presence or absence of these enzymes is kind of unique to species or even genuses, okay? So for instance, Staphylococcus versus Streptococcus. Both are common infections of the respiratory tract. Both grow in a pretty similar way. How do you distinguish them? Staph is facultative anaerobe. So it can use oxygen. Doesn't have to, but can. Okay? So when they use oxygen, they produce hydrogen peroxide. To neutralize hydrogen peroxide, they have an enzyme called catalase. Strep, on the other hand, is aerotolerant anaerobe, doesn't use oxygen, it doesn't generate hydrogen peroxide, so it doesn't have to have catalase. Catalase negative. And we're going to do that not next week, but sometime in the future when we're going to talk about gram-positives. You can distinguish facultative anaerobes and aerotolerant anaerobes by the fact whether they have or have not catalase. Good? Last little bit. So we're gonna add like hydrogen peroxide to the culture sample, and if catalase is there, you have bubbles. Okay, pretty cool. Very simple. There's a special term, capnophiles. They need CO2. Just like Helicobacter pylori kind of prefers. Campylobacter that we have here it likes it. Okay? So, Campylobacter, it's fine. It's gastroenteritis. It's not pleasant, to be honest. So, remember what we need to know about each pathogen, right? Transmission, whether there's a vaccine and the disease. So it's, diarrhea. Zoonotic can be from the food, like unpasteurized milk, eggs, poultry. Can lead to so-called Guillain-Barré syndrome, autoimmune myelitis, resulting in temporary paralysis. The only exciting fact about Campylobacter jejuni, in my opinion, is that the most recent outbreak it started in Ohio, and it was associated with puppy mills. That's it. Okay, it happens. It's not super common. Okay, the common cause of bacterial enteritis, but bacterial enteritis in the United States is really not super common. It's mostly viral. pH requirements, come on, taking candy from a baby. You have acidophiles, you have neutrophils, you have alkaliphiles, right? So optimal pH for acidophiles is like around 3. Alkaliphiles is around 9, 10. Acidophiles are more common, a lot of food that we eat contains actually acidophiles like lactobacilli or bifidobacteria. Human pathogens are neutrophiles. Okay. Acid or base, they screw up two main processes in the cell if you think about it. Remember, one process that depends on hydrogen ion concentrations, electrochemical gradient and their diffusion, flagellar movement, and another process is cellular respiration, ATP synthase. Okay. Temperature requirement. So, good news, don't memorize the numbers. Seriously, I'm not going to ask the numbers. I'm going to ask like qualitative questions. And I'm going to guide you through this. So, psychrophiles grow in the fridge only, period. If you're curious, if you want to see a psychrophile, get some Greek yogurt, eat it, like whole milk, no additives, whatever. Leave it in a fridge for a couple of months. You're going to have pink colonies, psychrophiles, probably leuconostoc. Okay? Now, psychrotrophs. So these guys grow at the room temperature or even at the temperature of the human body. But they can also slowly grow in the fridge. Do you see the difference? And they can spoil the food all right. So this is why they are the major cause of food spoilage. They can slowly grow in the fridge. Mesophiles, obviously boring because they can grow at the body temperature. Thermophiles, scalding water. Hyperthermophiles, boiling water. Does that make sense? Okay. Other factors. So, concentration. We talked about it before when we discussed tonicity and osmosis, stuff like that. Hypertonic environments are bad. But there are organisms that are categorized as osmophiles. So I want to correct myself. They don't grow in hypertonic environments. They grow in the environments with a very high osmolarity, like a lot of sugar or a lot of salt, that kind of stuff. So the reason like why I spent some time trying to find this picture for free, and it is free. So these are salt basins in San Francisco Bay. They produce sea salt there. You see the different colors? These colors are determined by the salt concentration. Because at different concentrations of salt, different bacteria and archaea are going to grow. And they will give different color to those environments. And basically they know, oh, okay, we got to that concentration because the color is green or red or whatever. Okay? I want you to draw a clear distinction between osmophiles and halophiles. Halophiles, the term refers specifically to high salt concentration. Salt, sodium chloride. Osmophiles, any chemical. So basically, every halophile is essentially an osmophile, but not the other way around. Make sense? Okay. Well, obligate means must. Barophiles, high pressure. Xerophiles, dry environments like deserts. Polyextremophile, my favorites, the most badass bacteria in the world. Deinococcus radiodurans. For some reason, someone decided to sample water from the cooling circuit around the nuclear reactor. I'm not kidding. And they found Deinococcus. If it would cause human disease, we would be doomed. It's unkillable. Like acid, fine. Boiling water, sure. UV light, not a problem. Gamma rays, well, you have to blast for like hours. The plates with Deinococcus were taken to the International Space Station and were exposed to the space. Taken back. These guys kept growing. I mean, they didn't grow in the space, but they're like, no, we're not going to die. So that's a pretty badass. Okay, questions? All right. So a couple more things here. I'll explain what I expect from you. Then we're going to wrap it up. We're going to do dilution problems, and I'm going to kick you out. Sounds good? Okay. So this. Basically, we just finished what I call the first chapter, concepts in microbial growth. Now it's time to talk about the control of it. Antimicrobial chemicals, methods, whatever. So, first of all, the distinction between the fomites and the living tissue. Well, fomite is anything, any object that is not a living tissue. Like your pen or your notebook or your glasses or, you name it. I want to make a small remark. Not air, not water, not food. Those are not fomites. But like my bottle is a fomite, but water in it is not. So two different types of protocols to use on fomites, to use on living tissue. Let's start with the fomites. Sterilization. You kill everything, including endospores. Am I clear? Like seriously everything. You can use chemicals, you can use physical methods like radiation or temperature, but you kill everything. Clear? Then we have disinfection. Endospores, probably not. Okay, so they're going to remain, but everything else is going to be gone. Again, you use your physical methods like radiation or temperature, or chemical methods. Finally, sanitization. For the most part, mechanical removal. So let me give you an example. If you take, let's say you have a dinner plate, dirty dinner plate. If you throw it into the fire and wait for, 10 minutes, this is incineration, and it's sterilized. Everything's burned. Okay, does that make sense? If you put it on the table and cover it in 3% hydrogen peroxide for 15 minutes, if there are any endospores, it's not going to get rid of them, so disinfection. Clear? If you take this plate and you wash it under the running water with a dish soap, sanitization, you mechanically remove bacteria from the plate. Am I making sense? Awesome. Living tissue. You cannot kill everything, period. Because, the methods that can kill everything will kill a patient. You can't throw a patient under the gamma rays, you can't throw a patient in autoclave, you can't burn them legally, so that kind of stuff. So we have antisepsis and degermation. Antisepsis. When you pump your hand sanitizer, spread it over your hands, and let it dry, this is antisepsis. Because your hand sanitizer, the chemical actively kills bacteria and enveloped viruses. That makes sense? When you apply chlorhexidine, that's antisepsis. What is degerming or degermation? Washing your hands. And if you think about the prep that surgeons go through before entering the operation room, they do both. They scrub, which is degermation, but they also apply chlorhexidine or betadine or whatever to provide antiseptic treatment. Is that clear? Cool. And that's it.